So for algae, both the chloroplast and nuclear genomes can be engineered, but depending upon what gene you put in, you can have very different outcomes. There are a variety of different ways to introduce DNA into the cell, but primarily for chloroplast, we do this by particle gun bombardment. So what that means is that we actually take our transforming vector, we coat it onto small gold particles, and then we shoot those into the cell with high pressure from gas. And we do that because for chloroplast, you have to get through both the outer cellular membrane as well as the chloroplast membrane in order to get that DNA into the chloroplast where it can transform the cell. So for algae chloroplasts, they have their own complete genetic system. So what does that mean? So first of all, they have DNA. Almost all of them are circular DNA. In this case here, we're showing the genome of Chlamydomonas reinhardtii, which is a little over 200 kilobases. And even though there are about 4,000 different proteins inside the chloroplast, they're responsible for photosynthesis, fatty acid metabolism, amino acid metabolism, most of those genes are actually nuclear encoded and transported into the chloroplast. And the chloroplast genome, in fact, only encodes about 40 or 50 proteins on its own. However, they have their own set of ribosomes, their own set of transcription and translation factors, tRNA, so they have a complete genetic system. The genetic system is pretty simple, and it looks very much prokaryotic in nature, and many people believe that's because the chloroplast actually came from a cyanobacteria many years ago. They have very simple promoters. Transformation proceeds by homologous recombination. That's important because we can precisely target genes. Proteins have disulfide bonds, and they're folded correctly, but they're not glycosylated. And importantly, we can get recombinant proteins to accumulate to relatively high levels. So in order to express any protein inside the chloroplast, whether it's a native gene or an introduced one, you have several requirements. First of all, obviously, you have to have a promoter. That's something that allows for that gene to be transcribed into an RNA. And then that RNA has to be translated into a protein, and that's generally determined by the 5' untranslated region, but there's also some influence of the 3' untranslated region. Recombinant proteins, as I'll show you, can accumulate to very high levels, and it's a kind of an interesting biochemical environment where disulfide bond and folding goes on, but glycosylation does not. So if we want to express a heterologous protein inside the chloroplast, not only do you need the gene of interest, you also have to have a selectable marker. That means you have to have a way to select the cells that have integrated in your recombinant gene or your recombinant protein, right? And the reason you do that is because it's actually a relatively rare event that you get the gold particle to get inside to a chloroplast and deliver that DNA. So maybe out of every million cells you have, only a few of those are actually transformed. So what we generally do is we have two genes, and in this case we show them head to head or head to tail, and you can put them into a variety of different ways, but this is one of the common ones. So you have a promoter, you have your recombinant protein of interest, then you have a second promoter, and in this case kanamycin resistance. That gives you kanamycin resistance so you can select the transformant that has an expressing your gene of interest. Primarily, gene expression is controlled by translation in the chloroplast. So what we found over the years is we can use a variety of different promoters to produce RNA, and that has very little impact on the amount of recombinant protein that accumulates. And what has a big impact on it is what 5' untranslated region that you use to express that protein. This is a picture right here showing the 5' untranslated region of the PSBA mRNA. This actually is a 5' UTR that confers high level of translation and therefore high level of recombinant protein production. And as I said, there are about 40 of these, and all of them give you a little bit different level of translation. And now we even have some synthetic ones that also work. We can actually accumulate recombinant proteins to relatively high levels. So what's shown here is two different transgenic lines called MA1 and MA22. So these are two different transgenic lines in which we introduced a recombinant gene encoding a bovine serum amyloid A3 protein or mammary-associated amyloid protein. So what's shown on the left is a stain gel of wild-type cells, and you can see these cells accumulate many different proteins. And then two transgenic lines, and I think in both of those you can see that the production of a new band there, and that new band is in fact the bovine recombinant MAA protein. And that is proven by the Western blot on the right, in which we've taken an antibody that is specific for that protein and showed that accumulation of this protein is at very high levels in those cells. We can also accumulate some mammalian proteins that require extensive folding and even assembly. In this case, what we did was we introduced both the heavy chain and the light chain gene of a human IgG1 protein. So this is an IgG antibody that requires 2 heavy chains and 2 light chains to assemble together. And here are a series of Western blots in which we show not only does the protein accumulate, so the light chain on the far left, the heavy chain in the middle, but if we run non-denaturing gels, as we have on the right, you can see that those proteins actually assemble into the much larger 4-protein complex, which is almost 150 kilobases large. And we were also able to show that these proteins, not only did they assemble, but they actually have binding to the antigen at exactly the same level as the mammalian protein. So relatively large, sophisticated proteins can accumulate inside the chloroplast. Now, not only do we want to express recombinant proteins, in this case either the MA or the antibody, quite often we're going to want to do metabolic engineering. So one of the questions that we've attempted to address is, well, how many different genes can we assemble into a single transgenic line? And one of the ways we decided to do this was to see if we could replace the entire photosystem 2 reaction center in a synthetic manner. So in order to do that, the first thing we had to do is go back and delete all of the native genes. So the reaction center of photosystem 2 is composed of PSBA, PSBB, PSBC, PSBD, PSBE, and PSBF. So all of those genes are spread out all over the genome as shown here by their letters. So the first thing we did was we went in, and because the chloroplast goes by homologous recombination, we could delete all those genes. Then synthetically, we assembled all of those genes into a single cassette. So we made the single cassette that's shown at the bottom, which had all of those different proteins placed head to tail, and then put that entire complex back into the strain in which we had knocked out or deleted all of the other genes, right? And what's shown on the left is transgenic lines that now have all of those genes strung back together in a new completely artificial cassette, and that restores photosynthesis. So in this case, we can show that we can make some very complex engineering inside the chloroplast, and it works quite well.